Epistasis is a type of polygenic interaction where one gene controls the phenotype of another gene for a trait. Both genes have an influence on the physical appearance of the trait, but the one that shows the epistasis masks the effect of the other.
The important thing to remember about epistasis is that the epistatic gene has no effect on the genotype or genetic composition of the gene being controlled. It affects only the phenotype. If the epistatic gene was not present, the gene being controlled would be expressed normally.
Epistasis is very similar to dominance, except that dominance entails the masking of genes on the same locus. So, epistasis involves genes on two different loci.
There are two types of genes involved. Epistatic genes are those that perform the masking on another gene, while hypostatic genes are those whose effects are masked. Just remember, one gene masks or hides the effect of another gene on a different locus.
In this video, we will be talking about dominant epistasis, recessive epistasis, and duplicate dominant and recessive epistasis. First, let's talk about dominant epistasis. Dominant epistasis is when a single copy of an allele is required to inhibit the expression of an allele at a different loci. When dominant allele B masks the expression of the gene at loci A, allele B is an epistatic gene that exhibits dominant epistasis. So let's use the summer squash as an example. Fruit and flower color in plants is a common example used to illustrate dominant epistasis. The squash has three possible colors. Yellow, big A, little a, is dominant over green, little a. So the yellow allele is dominant to the green allele. However, since the squash color is polygenic, gene B also determines squash color. Gene B shows dominant epistasis. Therefore, gene A will only have an effect on the phenotype of the squash if gene B is homozygous recessive. If gene B is heterozygous or homozygous dominant, gene A will be blocked, no pigment will be expressed, and the resulting squash will be white. You can see in the cross on the screen all of the boxes with a big B gene, the resulting squash is white. Only when there are two little b genes will color be expressed in the squash. You can determine whether a gene that shows epistasis is dominant or recessive by looking at the number of affected individuals in the cross. As shown, the majority of individuals, 12, will be affected and turn out white because they have a big B or big B little b genotype. The phenotype of gene A will only be seen if two recessive alleles of the B gene are inherited. 3 individuals will show the dominant big A or big A little a yellow phenotype, and 1 individual will show the recessive little a green phenotype. Remember that yellow and green squash must have a duplicate recessive little b phenotype because gene B shows dominant epistasis.
Okay, so I'm going to be talking to you guys about recessive epistasis. Recessive epistasis refers to recessive alleles at one locus masking the phenotypic expression of the other gene locus. The ratio for recessive epistasis tends to be 9:3:4. And an example of recessive epistasis is seen in Labrador coat color. So recessive epistasis is seen in the genes that determine Labrador coat color. These dogs may be black, brown, or yellow, and their different coat colors are determined by interactions between genes at 2 loci. One locus determines the type of pigment produced by the skin cells. A dominant allele, B, big B, encodes black pigment, whereas a recessive allele, little b, encodes brown pigment. Alleles at a second locus affect the deposition of the pigment in the shaft of the hair. Dominant allele, big E, allows dark pigment, black or brown, to be deposited, whereas recessive allele, little e, prevents the deposition of dark pigment, causing the hair to be yellow. The presence of the genotype little e at the second locus therefore masks the expression of the black and brown alleles at the first locus. The genotypes that determine coat color and their phenotypes are presented below. If we cross a black Labrador that is homozygous for the dominant alleles, big B, big E, with a yellow Labrador that is homozygous for the recessive alleles, little b, little e, and then intercross the F1 we obtain a progeny in the F2 in a 9:3-4 ratio as described earlier.
Talk about duplicate recessive and duplicate dominant epistasis. Flower color in the sweet pea is an example of duplicate recessive epistasis. Flower color is determined by both the A and B gene. A homozygous recessive genotype for either gene A or gene B will mask the flower color. If both gene A and gene B are homozygous recessive, the color is also masked. In duplicate dominant epistasis, a dominant allele at either gene A or gene B will mask the gene's effect. So duplicate recessive epistasis has a 9-7 ratio, while duplicate dominant epistasis exhibits a 15-1 ratio.
To review the information, epistasis is the gene interaction where one gene from one locus controls the phenotype of another gene for a trait located on a different locus. These examples mentioned before will help understand the differences between dominant and recessive epistasis.
Yeah, they masking and masking and masking and masking. They masking on genes. Yeah. Epistasis, big interactions with genes. One locus can mask the effects of a gene. Don't think same locus cause it's kinda different for these. One that does the masking is the epistatic gene. Other that is masked is the hypostatic gene. Never an effect on the genotype of a gene. Always gonna be the phenotype of a gene. Dominant recessive effects of these genes. Epistasis. One gene masks another gene, yeah. On a different locus, yeah. Masking by the epistatic gene. Hypostatic gene not expressed. Sometimes one gene masks another gene. Epistasis with the genes, yeah. And still, phenotype, it's a lot of phenotype that they masking and masking and masking and masking. They masking on genes. Phenotype, it's a lot of phenotype that they masking and masking and masking and masking. They masking on genes. Yeah, A.
Let's take a look at dominant epistasis. Check the squash example, yeah, it's really time to face this. 1231 dominant epistasis probably made this. Leaves a colorless squash, but no way is that tasteless. If it's recessive, you know that ratio changes. 9-4 tells us recessive is at play. Homozygous recessive keeps the other gene at bay. Labrador retrievers are the example of the day.
Epistasis. One gene masks another gene, yeah. On a different locus, yeah. Masking by the epistatic gene. Hypostatic gene not expressed. Sometimes one gene masks another gene. Epistasis with the genes, yeah.
Low dominance.
Triple X. 2 genes, this epistasis.
Young Chromo, it's lit.
ASAP
genes, we out!